Welcome to another edition of Flipped My Classroom. Tonight you'll be watching some information on the enzyme lab that we're doing on Friday. So the first thing you need to do is to open the lab. So you need to go to my website and go to the biology section right here. You need to run down here to under Lab Out Loud, Enzyme and Temperature Lab, and this will bring up the lab. Notice that it says here, even though you will collect data together in the lab, each student will write up a completely independent lab report. So you will need to copy this information and paste it into a new document. In class, all the materials are already set aside for you, and I have listed here some basic procedures. You need to wear aprons, you don't need to wear gloves, but goggles. And at your lab station there'll be a graduated cylinder, and you will pour 5 milliliters of hydrogen peroxide with 5 milliliters of distilled water from the squirt bottle. You'll take the cap off the vial that contains the pureed beef liver, which contains enzymes, from the beaker at your lab station and place it into the 125 milliliter beaker. I'll show you that in a picture in a second. You'll uncap the vial, insert a temperature probe, and get a temperature. Record this in your notebook. Using a transfer pipette, transfer 0.5 milliliters of beef liver puree into the biochamber. Then you will add 5 of the 10 milliliter diluted hydrogen peroxide into the biochamber. You will immediately cap with the O2 sensor and start recording the O2%. You'll do this every 10 seconds until it stabilizes. And you'll need to, notice here, swirl the biochamber in a consistent motion. So let me show you what the lab station looks like. Here's the lab station. This is your biochamber, and you see the O2 sensor on top there. Here's a vial. Your vial will have a little bit of pureed substance there. You have a transfer pipette there. This is a graduated cylinder. Here's your distilled water. Temperature probe, and here's your temperature and your O2%. So you will take this graduated cylinder, add 5 milliliters of hydrogen peroxide from the bottle, add 5 milliliters of water. You will uncap this, stick the temperature probe in there, get a temperature, record it in your notebook. Then you will add approximately 2 to 3 drops of this into this biochamber. Then you will add half of this graduated cylinder, the diluted hydrogen peroxide, into the cylinder also. Immediately you cap it with the probe, you record the percentage, then every 10 seconds you continue to record the concentration of oxygen. And at the same time you will constantly be swirling the bottle. Just pick the bottle up and swirl it around. So you're doing a motion like this. Now, in the lab, you will need to test 3 samples, of beef liver puree. So there will also be a sample in a hot water bath. There will also be a sample in the ice bath. You need to figure out the rest of the procedures for this lab. Everybody types up their own procedures. Everybody writes up their own lab report, even though you're working together. If you have any questions, we'll talk about it tomorrow in the morning.